Hallmarks of Aging. For a long time, there were 9 hallmarks were known for aging, and it was updated into 12 hallmarks in the year of 2023 in Cell paper. The first entity is the genomic instability. Genomic information is stored in DNA molecules, and over the aging process, this DNA structure becomes unstable. There can be single-strand break, or base mismatch, or double-strand break can happen. In normal cases, when the DNA is damaged, mRNA complex is recruited to damage the site. It further recruits ATM and phosphorylated. This phosphorylated ATM is activated kinase form, and it can phosphorylate H2AX. H2AX is a subtype of histone protein, and this phosphorylated H2AX is called gamma H2AX, and this is the hallmark of damaged DNA site. When DNA damage is properly repaired, this complex leaves the DNA and it can move on. However, in defective cells, the DNA damage is not smoothly moved, then the stalled DNA persistently accumulate gamma H2AX. In this case, they generate the genomic instability and inflammation. And the accumulated phosphorylated H2AX, so-called gamma H2AX signal goes up. The second hallmark is telomere attrition. At the end of a chromosome, there are some repeating sequence area, so-called telomere. And telomere is kind of a protecting groups for the chromosome structure, and this is composed of non-coding sequences. During the cell division, DNA should be replicated, and every step of replication, the telomere gets shortened. This is due to the direction of the new DNA strand synthesis, and Okazaki fragment can explain this shortening. Every step, the telomere getting shortened, and when it reaches the limit, then the cells cannot replicate anymore. So this experiment has been performed by Hayflick, so this limit is called as a Hayflick limit. In human cells, it is known that about 50 times division is a limit of cell division. The third hallmark is epigenetic alterations. When the embryo develops, in the beginning, near to the stem cell stage, the cells express almost every the genes, but when they developed into specific tissues, now chunk by chunk, big area of those genes are turned off. And this regulation is controlled by epigenetic patterning. So this epigenetic pattern can define the character of each organ, but this epigenetic pattern can be affected by habit or environmental impact. During the aging process, this epigenetic memory can be changed. For example, in normal cells, this area should be open form and gene expression should be turned on, but this area can be closed and turned off structure, or vice versa. So this kind of epigenetic altered state can generate a lot of disturbance to the life forms, and it can be the hallmark of aging. The fourth hallmark is the loss of proteostasis. Protein synthesis starts from DNA and through mRNA, and eventually the translation make protein. After that, proper folding is required to have the original function of the proteins. Translation is catalyzed by ribosome, and during this protein synthesis, there can be several errors can occur. Wrongly translated protein, or premature termination, or ribosomal stalling. In this case, synthesized protein is not released from ribosome, it can generate a problem. For the folding process, Chaperone is important the molecule to help the folding. If the chaperone function is somehow negatively affected, then misfolded protein can increase. This kind of the proteostasis breaking situation generate the problem of aging. In case of ribosome stalling, the synthesized protein is not released from ribosome, then the translational speed can be reduced. Also, this exposed protein is more prone to ROS damage, so as the result, the impaired protein can be generated. Now, chaperone can help for folding of this protein or assist to degrade the impaired proteins. If the normal protein folding is impaired, then it can go to the folded protein status. If not, the misfolded protein can be accumulated and generate aggregates. The fifth hallmark is a disabled macroautophage. Autophagosome collect the misfolded protein or dysfunctional organelle materials. But if there are too high load of these materials, the autophagosome capacity is saturated. It cannot handle all of them. Also, autophagosome need to merge with the lysosome, but the aged cell's lysosome has an increased pH. As the result, the enzyme activity is also reduced. So this kind of a lysosomal dysfunction also makes the problem for the macroautophage function. That's why altogether, the macroautophage, the efficiency drops, and this is one of the hallmark. The sixth hallmark is the mitochondrial dysfunction. Mitochondria has an electron transport chain, and this is responsible for oxidative phosphorylation. But when the process generates too much ROS, it can generate damage on the outer membrane of mitochondria. Then it can be the apoptotic signal and also can cause inflammations. So during this process, the mitochondrial, the DNA, can be secreted into cytosolic area too. The seventh hallmark is cellular senescence. In a sense, this is combined the different hallmarks in the cellular level. One is the DNA damage, also proteostatic stress, and mitochondrial dysfunction, and lysosomal the functional decline, and collateral the molecular damages in many different places inside the cell. The actively growing healthy cells go through cell cycle for the proliferation. Now, the cells are damaged, then the cell stopped the cell cycle, and it enters into quiescence state. But this quiescence state can be reversible. When they have the good signal, it can come back to the G1 state. However, if the stress is prolonged, now this temporary quiescence state can be changed into non-proliferative, the final stage of a senescence state. So senescence is the cellular level of aging. The eighth hallmark is the deregulated nutrient sensing. In normal status, if there is a nutrient is sensed, then this signal should go to the growth and proliferation because this is a good environment. If nutrient deficient environment, now this signal should activate maintenance and repair rather than growth and proliferation. So this maintenance pathway should be antagonistic to the growth signal. So this is a regular nutrient sensing system, but in aged cells, this system is disrupted and not proper signal is spread out and generate problems. The ninth hallmark is altered intercellular communication. Senescent cells generate the SASP phenotype. SASP is a senescence-associated secretory phenotype. When the senescent cell generates a lot of inflammation signals, it can recruit the immune cells. In normal status, these immune cells should clear out the senescent cell. But if there are survived senescent cells, they can make the immune cells exhausted, and as a result, this SASP does not affect the immune cells positively, but it can generate more the healthy cells and change them into senescent cells. So senescent phenotype is spread out through the aging process. The 10th hallmark is stem cell exhaustion. When the immune cells remove some of those senescent cells, those empty space is refilled by stem cell functions. So some cell can be self-renewed and then differentiated and make more cells to refill those empty spaces. But in the aging process, these stem cells are exhausted, then this refilling activity is reduced, then eventually the senescence phenotype is spread out. The 11th hallmark is chronic inflammation. If the senescent cell generates a SASP persistently, it can induce the systemic accumulation of pro-inflammation factors. Then the immune cell recruitment is also persistent. Then it can result in chronic inflammation. In healthy body, the microbiome should be balanced with a majority of probiotics with a minimum pathogens. The last hallmark of aging is dysbiosis. So in this case, the pathogen is actually surfaced the probiotic. This is not healthy environment. Why dysbiosis happens? Pro-inflammatory selection pressure may prefer the pathogens, so pathogens can survive better. And due to immunosenescence, the pathogen could not be completely removed and they survived. Also, epithelial cell and stem cell decay can be another cause.